Okay, I see that we are online already. So let me introduce you to the audience. So we are really pleased to welcome you, Paul. So we have Paul Avillach, who is Doctor of Medicine. He also holds PhD, and right now he's Assistant Professor of Biomedical Informatics and Pediatrics in the Department of Biomedical Informatics of Harvard Medical School. And he's going to continue our session on modern technologic solutions for practical healthcare with the talk on creating fair computational tool for the nationally scaled conduct of biomedical research. Paul, please correct me if I'm wrong with fair, if it's not correct pronunciation of this abbreviation. Okay, please, the floor is yours.
Thanks, Daria. So thank you very much for this opportunity and to invite me to your conference. And so I'll be giving you an overview of the various open source software that we have been creating within my lab at Harvard Medical School to enable to integrate very large scale clinical and genomic database to be able to discover new subgroup of disease. So within my lab, half of the team are software developers and half of the team are postdocs and students that actually analyze the data using the tools that we are creating. The whole concept of what we are creating is taking the analogy of Google Map, where we have all the different layers of information, satellites, images, postal code, traffic jams in real time. We all use this in production on our iPhones today to be able to have all those different sources of information, everything centered by GPS coordinates. And what we have done is to create the same system and creating information common organized around individual patients with all the different layers of information, with the exposome, the clinical data coming from registry, coming from electronic health record, the genomic data, sequencing data, gene expression, any kind of clinical or genomic or environmental data being centered into the same system, the same database. And that's something that we created called Picture. It's an Apache 2 license open source project that enables to bring many different data types at scale. And that's one of the key elements, because at first we were using, in the context of the big data in health, could be on 3 different axes, either by the number of patients or the number of variables per patient. For example, when you have all genome sequencing data, you definitely have a lot of variables for each of the patients. And also when you have deep phenotyping with deep registries with more than 100,000 variables per patient, or the full clinical notes of patients within an institution. So you can have all those different variables, clinical or genomic. And the third axe is time, because we are all living entities that change at every single second, and we can, for some system, record many of those measurements over time. So we needed to have a system that can scale in 3 different axes: number of patients, number of variables, and including all genome sequencing data, and time, including all the different time. And that's why that's the principle of how we created Picture that enables to scale. Another very important principle was also to use the cloud, because there's no way we can today, for example, the top med studies, and I'll talk about it later, where from NLHBI, the National Heart, Lung and Blood Institute from the NIH, with the largest clinical and genomic cohort that could be analyzed on-prem, on servers in clusters within institutions. That's why we have to now use the cloud. And by integrating all the different clinical genomic data and the imaging and in the cloud, so that the data is next to the analytics in the same cloud environment to reduce the costs, and into very secure environment that enables for an investigator to actually run his own analysis, but also to share the data with other investigators, where they bring the analytics next to the data. And that's how we've been also expanding based on using in the cloud. Showing you, and so with this model of using picture is now on more than 1,400 projects currently in the lab. And one of them, Biodata Catalyst from NLHBI, has more than 3 petabytes of data, 240,000 participants, 158,000 full genomes that are included within this environment that can be queried in real time. And so we have more than 80 studies from the TopMed Omics Precision Medicine, including the CRAM and the annotated BCF, into this environment with next to all the detailed clinical data that enables to search and extract all this information. And this model of picture is being deployed in all those various projects by Data Catalyst, also at the Broad 7 Bridges Genomics, on the UDN Disease Network, also internally at Quest Diagnosis, the Boston Children's Hospital Biobank, where now we have 2.9 million patients, clinical and for the more than 20,000 consented patients with the sequencing data available, the GIC network across all those 5 pediatric institutions, and so on, in the context of having, so we use the same tools, application, open source, so that anyone can use them across and to be able to integrate at scale clinical and genomic data. The way we organize the architecture is in picture, high performance data store, which is a variant store that enable to scale with all the longitudinal clinical EHR data. So that's a variant store that we created where we load the annotated BCF into picture high performance data store with all the clinical data, EHR registries, with all the sequencing data into the same database. And then this database can be queried via the picture API, either by a search portal where you can type any clinical or genomic variable of interest to be able to narrow down your cohort, and also to extract in R and Python into any analytic portal. It can be using Terra, 7 Bridges, DNA Nexus, or Galaxy, or any workspace environment can extract the data from picture in R or Python. And a new analytic portal that we created in co-development with Amazon, Service Workbench on AWS, is another open source project, and I'll talk about it later. So the whole spectrum of what we're creating and available throughout all our projects is picture to store and search and extract the data, and Service Workbench to actually analyze the data. And showing you a screenshot of how the user interface of picture looks like, where you can query any of the clinical data with Boolean queries. For example, all the patients that are male with a genomic variant on the gene CHD8, the number of participants, and then you can click to be able to see the pop-up window with the annotated BCF coming up in real time, so you can narrow down your cohort and export the cohort that you're interested in. The user interface is really to be able to search, extract, create your cohort, but then you will be running the analysis into an analytic portal. And that's where we provide many, many examples of also publicly available in GitHub with how to access and extract the data using the picture API in R and Python, with many different already written examples based on the various dataset that enables you to get started and jumpstart to run the analysis, to be able to run it at scale. And so those examples show you with where you provide the authentication and authorization token, everything is explained based on the dataset that you are authorized to access, to then be able to run and directly have an example of how to run your an analysis. And then, because it's a Jupyter Notebook, you can definitely add your code and change the existing code to be able to run your work. And for all those different datasets, we're providing a data access dashboard to know for all those different cohorts for example, with picture from Biodata Catalyst, of the various studies that have been integrated, the number of clinical data, the number of phenotypes, the number of sequence variable, so that you can know what is available today behind the interface for you to be able to analyze. So what I showed you so far is here the search portal based on the user interface of picture, of how to extract the data using picture with R and Python clients with Jupyter Notebooks. And now the last bit I wanted to show you is the analytic portal service workbench on AWS, which is a new open source software that we co-developed with Amazon. And this started 2.5 years ago now, where discussing with AWS engineer, where they looked at the Terra platform from the Broad and many other platforms like Seven Bridges Genomics, like DNA Nexus, and all of them were not open source and were not available for you to be able to install. Terra from the Broad on Google is open source, but you can't install it yourself on your environment under your security perimeter. So what we wanted is to be able to create from scratch a new workspace environment that would use the scalability infinite from Amazon of being able to run the different workspaces. So looking at taking the best of all those different platforms and workspaces and creating an orchestration with a very nice user interface that used the whole power of Amazon. And the purpose was to democratize the access to public cloud computing for the context of teaching and research with, and something very important, a full billing control, because it can be very expensive in the cloud if there's not safeguards based on being able to automatically stop various workflows that are running. And that's how we, for example, have imposed that by default, any workspace that is being created, if it stays in an idle time with nothing being run for 60 minutes, it will automatically stop, and to avoid the cost. Of course, if something is running, then it won't stop, but the default that can be changed as you wish when you use the platform enables to avoid astronomical costs for someone that will forget to stop an environment. And all this into very secured environments. And so on the drawing board, asking AWS engineers, where we wanted to be able to manage multiple funded projects for multiple courses and also for teaching across multiple Amazon accounts, centrally managed, something very secured and nice user-friendly user interface. And they managed to create it. So we had a first pilot project in our department at the BMI at Harvard, but now it definitely scaled, and now it's a full Apache 2 license project. It took 1 year of negotiation with Amazon lawyers, and they approved that it's a true Apache 2 license and not an awkward Amazon license. So everything I've showed you so far is truly open source Apache 2 picture and service workbench, so anyone can reuse it and add it within your own project with no IP issues, no regulation issues and all that, because you can have it and deploy it within your own environment to be able to scale. And the condition also was to have something extremely secured with patient data. So we reach HIPAA compliance, working towards now FISMA compliance with service workbench, where we have FISMA is a very high level of security in the United States that enable to be allowed to host federal data on behalf of the US government. And that's and moving Service Workbench into the FISMA world. That's ongoing work right now. And one of the key elements is the fact that Service Workbench is not hosted on the R and my GitHub account anymore. This is not a pilot project. It is an official AWS solution maintained by Amazon, which makes it much, much more powerful, much, much scalable. So I don't control Service Workbench. It's controlled by Amazon. But so this was a pilot project within the lab, but now there's 8 full-time developers from Amazon maintaining this solution, truly scalable, and that anyone can use it's free. The only thing you pay is the usage of the underlying infrastructure, of course, but there's no additional cost like you would have in many of the proprietary platforms to launch your different workspace environment. And so that's something that is a revolution in the context of this is the very first true native cloud workspace environment that is open source, that can be used and scalable. And it works because it's using the underlying AWS technology. What was created is a very nice user interface for an end user to be able to launch those various environments in less than 5 clicks of a mouse. Someone can launch a compute environment, for example, a virtual machine, or a notebook with Jupyter notebooks, Jupyter Lab with Python and R kernel using SageMaker, using RStudio, or distributed compute with an Elastic MapReduce cluster, where, for example, there's Health Variant Store that was developed at the Broad Institute. So the Health Variant Store that was used to create the Nomad database can be launched in just 5 clicks of a mouse from the Service Workbench. So you don't have to install it yourself. You have a nice user interface, and the underlying is using the development from AWS to have something at scale. And the key is there's no limit of those open source contributions. So anyone can add, and there's a growing community of creating those various workspaces. For example, we're now testing IGV. So IGV can be, and it's in a dev environment, can be launched within this by just a click. And then, so you don't have to install it on your environment. You just click and automatically it will launch as needed, and then it will stop. So the cost is very minimal to directly connect to your BAM file to be able to check the sequences at scale. And you can very easily choose the size of your environment. And so this dual environment of having picture and Service Workbench, that's what we use in many projects, including, for example, the Gic Project, Genomic Research and Innovation Network, across 5 pediatric institutions. And looking at the data from Boston Children's Hospital, Cincinnati, and CHOP, the 3 first hospitals, we now have within this network 5.4 million patients with 50,000 samples using this picture to search and query the clinical and the genomic data at scale, and Service Workbench to actually run the workflow environment across the different projects. And a new project that was just awarded yesterday, there's a public announcement, Aim Ahead, on Artificial Intelligence Machine Learning Consortium to Advance Health Equity and Research Diversity in the United States, of how to help health disparities and inequities across the United States, across many different sites. So it's a new project. It's a $100 million project over 2 years that will definitely leverage the infrastructure that I just showed you using a picture and Service Workbench, and that definitely will scale across the United States. And so that's out of the press. It was just announced yesterday, and happy to answer to any questions about those open source tools that definitely can be used within your own projects very easily, and has proven to scale across, and that's the key in scaling across the 3 different axes to make sure that all the various use cases could be answered. So I don't know how it works to answer to questions. Daria or Vladimir?
Yeah. Hello, Paul. I'm not Daria, but I will try to ask some questions. You made a huge system, and we are also building something like that here in Russia. But our storage is more humble because we are building 1 petabyte with 10,000 full genomes. And we also incorporate genotype with medical cards. But your system is is what we wanted to build 2 years later. So I guess later on we will talk privately with my colleagues who really want to talk with you. And as for one,
happy to. And the key is it's an open source project. So I mean, it's you happy to help, but that's something that you could use, explore, extend as you wish. It's on an Apache 2 license. So that's the key.
Yeah, I understand. And where do you store the genomes within this system on AVS from the biobanks? From, for example, Boston Biobank, or Boston also gave you API and you retranslate this API to open source. How does it work?
So we picture. We store the data into HPDS, the high performance data store, which is a Java-based application and stored in text file in a special format that is ready for query into a very, very efficient way. The key is it's extremely fast and scalable because there's a user interface. So when you query all those genomes, it has to be extremely fast because from the user perspective, we can't wait 10 minutes to answer. So that's where we store. Now, the implementation, do you need to use the cloud and Amazon for picture? No, you don't have to, because it can run on any virtual machine. So any virtual machine, it doesn't have to be in the cloud. For example, across those different institutions, Cincinnati is the on-prem instance of picture is on-prem on their hardware, well, virtual machine behind their firewall. At CHOP and Boston Children's Hospital, it's on Amazon. So the picture can work on Amazon, like any other VM, or on-prem. Service Workbench on Amazon can only run on Amazon because it's using Amazon resources. So that's definitely, it has to be on Amazon. You can't run it on Google Cloud or Azure. But so those are trying to answer to your question of where those things can run.
Yeah, and what about, you say, own text format? It's close to FASTA, or it's close to BAM, or it's close to VCF format? It's
closer to VCF, because what we load in HPDS is not the BAM file, is not the FASTQ file. What we load here to query against the clinical data in real time, it's the annotated VCF. So it's much smaller. So what is being stored in HPDS, so we have all the FASTQ file and BAM file up front, but what is loaded in the same database as the clinical data, it's the, because when you make a correlation between clinical and genomic data, you won't do it on a FASTQ file or BAM file. It's mostly on, except there's some exception, but it's mostly on annotated VCF. And that's why we load the annotated VCF and the complex EHR, longitudinal or registry clinical data in the same variant store. And the so it's closer to an annotated VCF into a specific format so that it can scale also on the free access with the time, because the time is also very important when you have EHR data.
And as for medical data, is there only diagnosis or some analysis that, like blood pressure, sugar, or something?
Anything, any clinical data, including clinical notes. We load any kind of clinical data from the EHR. No limit, nothing. This picture was created using the model of I2B2, so it's the grandchild of I2B2 that scales in the context with full genomes. So any kind of clinical data can be stored in picture. That's why we created picture. So the diagnosis, lab values, medication, sleep waves, EKG, any kind of clinical data, because we have the time. That's why in the database model, we store on the three axes: number of patients, number of variables, and time. Time is crucial. When you just have sequencing data, you don't sequence every day, too expensive, and there's no point because it doesn't change every day. But we have sequencing data, and sometimes we have multiple samples per patient, across different days, or in the context of tumor, we can have multiple samples over time. But for the clinical data, the time is crucial. So that's why we store per the second of based on when the data was collected in the same database model as the variant data, so that everything is already in the same database. So the query is much, much easier and faster because it's in the same data format.
Is there underlying some kind of NoSQL database like Mongo?
No, we tested many different variant store at scale to try to have those three. And at the end, we created from scratch picture and the high performance data store, because none of the existing database system would scale in the context of thousands of genomes with thousands of clinical data at scale. So we created something from scratch, publicly available, HPDS, picture high performance data store, because we tested more than 20 different variant store and clinical store with the longitudinal EHR data, and there was nothing that was scaling. So that's why we had to create a new one, which is the bad solution of creating yet another system. But because none of the other ones were scaling based on the data that we currently have. Today, we haven't reached a limit of, we didn't find a data set that was blocking us from scaling. So 158 full genomes with the full annotated VCF from TopMed is integrated with all the clinical data at scale, and they didn't put the system down yet. Will it scale with 1 million? We don't know yet. We don't have access to the data. And so, hi, Dana. And so it's that's why we created this new database system.
So you say that it's convenient to mix up some different types of data, analysis, plain text, diagnosis. It's okay, you can deal with it somehow, because
yes.
Here in Russia we have problems to normalize some data, because even for diabetes first, diabetes second type, sometimes doctors only print diab, and you don't know is it first or is it second.
And we have the same problems. It's what I'm saying is this. There's no magic here, to be clear. We are just storing all the data as it was collected, and then it's at the time of analysis that you create your computational phenotype of how, based on your research question, how you want to interpret having first diagnosis of diabetes versus the second one. Are you going to use HbA1c lab values, the result of it, as a way to be able to say if there's diabetes, or even in the context of diabetes, has HbA1c has been tested? Because if it's tested for a patient on a regular basis, even without getting the result, there's a very high likelihood that the patient has a diabetes. So there's all those different ways of analyzing and extrapolating the data will depend on your research question. Here, what we do with picture is to enable to put all the data into one place so that you can make those different analyses as you wish when the time of the analysis comes, because this will change based on your research question.
Yeah, thanks, Paul. I guess it's time to continue with Dana Wiesman. Daria?
Yes. Actually, I wanted to ask one last question to Paul. I think I have 1 minute. Donna, sorry, we will move to you. So, Paul, what is the place of community? What is the value of community you see for your project? So, I've heard that you mentioned that community can create images for environments. What else?
Yes, and also in the co-development, that's what we do, of different features to expand picture. So there's many different areas based on, and the way it works is like, this is open source project. Anyone can come and take, and there's anyone would want to adapt a bit based on the needs of their own project. And then that's why we have a community, the co-development of creating those new features. So there's many different opportunities that are ongoing across those different projects.